Um, so my name's Carrie De Jesus. I'm a PhD candidate at the University of Florida, and today I'll be giving you my talk on reptile host of Ixodes scapularis. What role do reptiles play in the epidemiology of Lyme disease? So here in the United States, Lyme disease is typically thought of being located in the northeastern United States, but it is present in the southeastern US as well. And the ecologies differ between the northeast and southeastern systems, specifically in the hosts that are available to the ticks. So in the northeast, primarily the white-footed mouse is a really good host for the ticks, as well as the Lyme disease bacteria, Borrelia burgdorferi. But in the southeast, the white-footed mouse is not present, and it's been suggested in the southeast that reptiles are a very important host species for the tick life cycle. So just so you can kind of see where Lyme disease is distributed across the US, as I was saying, the Northeast is where the majority of cases occur. We're also getting more clusters in the northern Midwestern US as well. But you can see down below in Florida, we do get reports of cases throughout the state. And taking a closer look at the state of Florida, this is the number human case incidence. So the darker colors indicate Lyme disease cases acquired within the state of Florida. In dark blue is where we have more cases, and the lighter blue is fewer cases. But you can see the northern part of the state has a higher case incident versus the southern part of the state, but we still see some cases down south as well. So when we're thinking about vector-borne disease and host relationships, we want to talk about pathogen competence. So you kind of need these couple different factors for a pathogen to persist in the environment and its host species and a vector species. So we need to know that the host can be infected with the bacterium. We need to know that the vector, in this case, a tick species, can be infected with that same bacteria. The host and vector, independent of one another, have to be able to support that bacteria. But we also need to know, do the host and vector interact at all? So if the pathogen is present, that's great, but we need to know, are the host and vector interacting? Because if the host isn't interacting with the vector, that pathogen can't be passed to and from, kind of stopping the bacteria cycle. So reptiles are a known host species for Ixodes scapularis, the black-legged deer tick, which is primarily the vector of Lyme disease. And so previous studies found in a laboratory setting that green anoles and southeastern five-line skinks were competent for Borrelia burgdorferi, the Lyme disease bacteria, but this hadn't ever been tested in the field really, and Florida is one of the most diverse states for herps in the US. And so I was interested to see if any other species could be playing a role in the epidemiology of Lyme disease. So the questions we wanted to address in the study is which species of reptiles in Florida actually interact with Ixodes scapularis? How common is it for these ticks to interact with reptiles? And we looked at this using museum specimens from the Florida Museum of Natural History and the Archibald collections. And then what is the prevalence of ticks on reptiles when we kind of test this idea out in the field as well? So we looked at 11 native Florida reptile species and examined over 5,000 museum specimens. So each specimen is one individual lizard, and they dated from 1902 to 2018. And this covered the majority of Florida counties. And then here's just kind of an idea of what the ticks look like on the reptile. So on the left-hand side, we have a skink with some nymphal Ixodes scapularis that are engorged. So you can see that they're kind of filled with some dark color, which is the blood. And on the right-hand side is a museum specimen where you can kind of see these dark and yellow areas, which are larvae that have gotten up underneath the scales of the lizards. So using our museum data, we looked at anoles and Plestiodon skinks, since those were tested in that original laboratory study for the competence of the bacteria infection. So looking at all the anoles, so over 1,200 samples, we didn't find any anoles with ticks. But when we looked at Plestiodon skinks, which include the southeastern five-line skink, common five-line skink, and broad-headed skinks, we found quite a few that we had a good distribution of samples. And here's our table with what we found on the individual host species for ticks. So I was saying anoles didn't have any ticks present, but the 3 species of Plestiodon skinks commonly had ticks. And so of our broad-headed skinks, about 25% prevalence. The southeastern 5-line skink, which is the species present in Archibald, had about 16% prevalence, and our common 5-line skinks about 18%. So with our museum data, kind of knowing what species we could target in the field to collect ticks to test them for the bacteria, we went to 6 field sites across the entire state of Florida. So 2 northern Florida field sites, 2 central Florida field sites, which included Archibald Biological Station, and then 2 South Florida field sites. And so in order to collect the ticks from the lizards, we used a technique called lizard fishing, where we would take a pole with a mealworm or a cricket attached to the end to lure the little skinks to bite onto them and then put them into a bucket and examine them. We also set out drift fence arrays to catch lizards as well. And then if we were able to capture them by hand, we also did that. And then we identified all the lizard species in the field and took tail snip, blood, scale, and tick samples if ticks were present on the lizards. So from the species we were able to capture from our field studies, we found that the southeastern 5-line skink had about 23% prevalence of Ixodes scapularis on them, while the common 5-line skink and the broad-headed skink had a much higher prevalence. We did catch some glass lizards at one of our central sites, but they did not have any ticks. And so this is kind of just a quick look at how many ticks we found per the animals and versus males, females, and juveniles. We didn't see any differences in males and female tick infestation with the nymphs, and we didn't see any significant differences between species and tick infestation as well. So we also wanted to compare our lizard tick hosts and see how they were distributed across the state of Florida. So we found that the northern part of the state had hotspots for lizards with ticks present, and we did this for all lizard species. So we had the hotspots in the northeastern Florida, and then it kind of slowly extends in the South Florida. But when we look at just the Plestiodon skinks in general, we see a much stronger pattern and hotspot for ticks on reptiles in Florida. And we then compared this to human cases. And so we saw that we kind of see that that northern part of the state has the higher case incidence, but we don't see too many human cases in South Florida, but it indicates with our reptile ticks on Plestiodon skinks that we do have a little bit of explanation on why we may be seeing some cases in Southern Florida. And we're still working on a way to kind of statistically analyze and compare human case incidence to our hotspot data with Plestiodon skinks and their ticks. And so just kind of wrap things up, we found that the Plestiodon skinks were a really good host of Ixodes scapularis in Florida. A couple of the ticks from Archibald themselves came off of some samples taken from the station. So we had 4 southeastern 5-line skinks from Archibald that did have ticks on them, which was exciting. Museum and Field Specimens provide insights into host usage and distribution. So by using field data and museum data, we're able to get a good look at how lizards are being used in the state of Florida and the relationship with ticks. It appears that lizards with tick distribution somewhat aligns with human case incidence, and we're still working out a way to statistically analyze that. And we're currently working on the molecular testing for the lizards and the ticks and testing for that Borrelia burgdorferi bacteria and other tick-borne associated bacteria and see if we find any infections present in the animals or the ticks themselves. I would just like to thank everyone who allowed this field research to happen this summer, including Betsy and everyone at Archibald for being very generous for letting us come out, especially during COVID, and all my field techs and everyone involved in this project. And now I'll take any questions.
Thank you very much, Carrie. I do have a question from Clara Mullins. She wants to know where your northern sites were located.
They were located at the Tall Timbers Field Research Station in Tallahassee and Big Talbot State Park. And Big Talbot State Park was very ticky. Pretty much all the lizards we caught there had ticks on them.
Great. Well, that's all the questions that I have for you right now. I think one might just come in. Yes, I do have another question for you from Jenny Schaefer. Do you have any hypotheses about why skinks are better hosts than for the tick than anoles?
I think the skinks are down in the leaf litter where the ticks, these Ixodes scapularis, tend to hang out in the southeast. And also I think the way their scales are positioned on their body, it allows for a little bit of a gap to reach the skin, and the anoles kind of have almost like little bubbly scales is the way I would describe it. Why you can kind of see on the screen here, you can kind of see the separation between the scales, where it's harder to see that on the anoles. So I think it's just easier for the ticks to attach as well.
Okay. And then we have another question from Hilary Swain. What other tick-borne diseases are associated with reptiles?
So we know that reptiles can have, besides Borrelia burgdorferi, other relapsing fever Borrelia have been detected in South America in reptiles and ticks. And then there's some work ongoing on different Rickettsia species and Ehrlichia bacteria that are slowly being discovered, but we don't know a lot about ticks and pathogens associated with reptiles. It's pretty poorly studied globally. So it's kind of hard to know, but that's why we're doing the study.